almost done. Is everybody excited? Yeah. Sure? Not really? Really? I would expect everyone to be very excited. All right, so we're going to finish up the material that we started on gastric function. And then we're going to jump into lecture 21. Okay, so lecture 21 is pretty dense. It's basically your intestines. Okay, so since it's covering all of that material, we're going to jump right into that. And if it turns out that we don't have time to get into the liver, we're at least going to touch upon some of the liver content in the intestinal lecture. So we'll kind of play it by ear to see if we can get through everything. The last part that I left off with was talking about gastrin. And I put this figure up to remind you how gastrin and CCK are related to each other. Gastrin is a very potent stimulator of secretion of what in the stomach? Gastric acid, right? So hydrochloric acid is going to be released in response to gastrin. It's not the only stimulator, but it is by far one of the most strong stimulators. You have two different forms. You have little gastrin, which is found primarily in the G cells in the antrum of the stomach. It is more potent. It does paracrine action there, and it's quicker. The duodenal version is mainly big gastrin. So the difference really is just size. They still have the same active part on the carboxy terminus. Big gastrin is not as potent a stimulator of acid, but it still is stronger than your histamine. The big difference here is that it is metabolized more slowly. It has a longer half-life in the blood, so it sticks around a little bit longer. So little gastrin does the job. It's a quick and dirty way to ramp up acid secretion. Big gastrin released from duodenal cells is there really to help prolong the effects of gastrin. Gastrin is going to be directly inhibited by somatostatin. So we haven't started talking about somatostatin yet. Do you remember when I mentioned you had neuronal, you had histamine, and you had gastrin, all of which are going to promote acid secretion? And I said that I had a nice big figure that would show how everything was related. That's what's coming up next, okay? So, if it ever changes, it's online. Okay, so first thing I want to start with is talking about what we're looking at here. On the top, those top two panels, so let's see if it will line up for me. This one and this one are both corpus. Down below, we're looking at the antrum. So this is all stomach, okay? We're looking at what's actually happening. On the left-hand side, those are your parietal cells, okay? It's the lumen of the corpus. These are your acid-secreting cells. They're also going to release your intrinsic factor as well. They're going to be ramped up not only in secretion, but also in proliferation in response to mediators that promote the release of gastric acid. On the right-hand side on the top, where you see the ECL cell, that's part of the lamina propria. So it's not directly in contact with the lumen of the stomach, but it is directly adjacent to the cells that are in contact with the lumen. So it's not going to sense the content directly, but it will be able to release the different mediators that will stimulate the parietal cell to cause acid secretion. Okay, so the first thing I want to point out is this parietal cell on the left. So when we're looking here, parietal cell here, okay, what's the box around? That is your, anybody know? That's going to be for your somatostatin. We're talking about D cells here. This is the one that we haven't talked about yet. So far, we've talked about your M3 receptor for acetylcholine. That's coming from parasympathetic postganglionic nerves. You have your H2 receptor on the parietal cell that responds to histamine. And you have your receptor for gastrin, your CCKB or CCK2 receptor. All three of these exist on the basolateral side of your parietal cell. And all three of them are going to stimulate that parietal cell to secrete acid. With me so far? Okay. Somatostatin is a receptor that's located in the same spot, but its job is to inhibit acid secretion. On the top right, in the lamina propria area, we're looking at ECL cell and also looking at a D cell. The big part here that's important to note is that this ECL cell is also going to be inhibited by somatostatin. Somatostatin's job is to shut down acid secretion by all possible avenues, okay? At least endocrine avenues. Your ECL cell, that's your enterochromatin-like cell, is releasing histamine, which will bind to the parietal cell on the basolateral side to cause acid secretion out the apical side. If you want to stop having acid being released, what's something that you do for that? You shut it down with the exact same mediator. So even though you have 3 different ways that you get the parietal cell to be stimulated, and gastrin, remember, doesn't just stimulate the parietal cell, it also stimulates the ECL cell. See here, we have a gastrin receptor here, we have a gastrin receptor there. Okay? We also have a gastrin receptor here. When gastrin's released, it stimulates acid secretion, it stimulates histamine secretion, then histamine also stimulates acid secretion. But the other thing it does is it causes its own negative feedback. It feeds back on these somatostatin cells. The D cells are going to reside in the corpus and the lamina propria and in the antrum of the stomach. What's one thing you notice about these 2 D cells that makes them a little bit different from each other? So one D cell is here, the other D cell is here. Say it again. So they'll both have gastrin receptors, in order to be able to to stimulate somatostatin release to feed back on itself. It's just not shown for size. But in terms of the actual cell structure, what's different about these 2 cell structures? Where are they located? One of them is in contact with the lumen. One of them is not. Okay? So the big question is, the difference between these 2 D cells in the corpus versus in the antrum is how somatostatin gets triggered to be released. Yes, gastrin will bind. If it's released from antral G cells, it will bind to somatostatin D cells and cause somatostatin to be released in order to shut down its own gastrin secretion. But it doesn't just have to be a neuronal or a hormonal signal. The antral D cells also can sense pH directly. So when there's a lot of acid being released by parietal cells, your corpus D cells aren't going to be able to sense that. They're going to secrete somatostatin in response to neuronal triggers, primarily. They will respond to hormonal signals as well, but they will not respond to an increase in protons present in the lumen. That's reserved for the D cells that are found in the antrum. So the biggest difference is even though you have D cells throughout the stomach, only the D cells in the antrum are going to be capable of responding to a massive drop in pH. Okay, so if something were to happen to the neuronal signal that's going to the corpus, you may or may not get that response to shut down gastrin release. But as long as the pH drops in response to acid secretion, the D cells in the antrum will respond. And they will release somatostatin to shut down more acid secretion by way of shutting down the avenues of hormonal secretion, gastrin release, histamine release, and directly onto the parietal cell itself. So the red box that I had up here in the beginning, that's your direct way to get somatostatin to shut down acid secretion. I'm going to trace the lines just so you can see them, but I will erase them because I want you to actually be able to read this. So when somatostatin gets released from here, it can enter an endocrine to bind to its receptor. You also have paracrine inhibition from somatostatin in the corpus. Okay, both of those will directly act on. Both of those are going to act directly on the parietal cell itself. The indirect somatostatin receptors are going to be to shut down gastrin and histamine. So 3 different cells involved in stimulating and also releasing acid. 1 hormone is responsible for shutting it down, and it acts on all 3 avenues. It stops the parietal cell directly. It stops the G cell from secreting gastrin. It stops the ECL cell from releasing histamine. Okay, so somatostatin is sort of the key player to stop acid secretion, to inhibit acid secretion. Can CCK also inhibit acid secretion? Yes. Why? It's related, right? It's related. And the CCKB or CCK2 receptor has basically equal affinity for gastrin and CCK. It's just that you usually have a lot more gastrin present in this area than you would CCK. But if CCK is floating around your bloodstream, it doesn't mean it can't also have an inhibitory effect. So the next question I wanted to ask before I have a summary slide is really just to see if you can reason through which one or more of these would be a method by which you can inhibit acid secretion in the stomach. You guys can talk to each other. What would be the effects of each one of these? So that's still good. The majority of the people that responded, we still have more people that got it right than didn't. So acidification of the antrum. What's going to sense that? That's going to specifically stop acid secretion. Should qualify that. The D cells in the antrum. Remember, the antral D cells are sensing the drop in luminal pH, and somatostatin is released in response to that. Somatostatin is released from D cells in the corpus because of neural or hormonal, not directly sensing that luminal pH. Okay? What about acidification of the duodenum? Well, so acidification is going to be sensed by the S cells to release secretin, to get bicarb coming out of the pancreas. But what else is present in duodenum? In relation to triggers for acid secretion. We have a little one and a big one. Big gastrin is produced from duodenal G cells. So it's actually going to help to shut down gastrin, big gastrins present in the bloodstream if you acidify the duodenum, because it will inhibit the release of gastrin from those cells. Blocking histamine receptors, that's a direct effect from corpus D cells. That's going to stop histamine from being able to have that paracrine activity, and then obviously directly inhibiting the gastric HK pump would be another way to shut it down. What is the main or most widely used medication to block the HK pump? Anybody know? Say it again. Prilosec, right? Your HK pump is a proton pump. So you've heard of proton pump inhibitors? Prilosec, which that's the brand name, but omeprazole is the generic. We talked about that once before in relation to lobbying. Omeprazole will directly inhibit that gastric HK pump. It doesn't really have much of an effect on the colonic HK pump, despite the fact that they have similar function. Okay? So all four of these actually will result in a reduction in acid secretion. They will do so by varying degrees, but they will all slow down the release of acid from the parietal cells. So this is really just to kind of summarize what hormones are involved and how they all can act to inhibit acid secretion. A lot of them will act in an endocrine fashion. Some of them will be able to act in a paracrine fashion depending on where they're present. All right. Now, the last part I want to talk about is sort of recapitulating the cephalic, gastric, and intestinal phases of digestion, but this time specifically in respect to how they result in changes in stomach acid secretion. So the first one is cephalic. Cephalic is going to be triggered by what? What are the mediators? Is it a hormonal mediator? Is it a chemical mediator in terms of like actual nutrients or changes in pH? Is it a neural mediator? What's going to be involved in the cephalic phase? It's all neural. All of it is neural. The cephalic phase, by definition, is the phase of digestion that is initiated by the nerves, by your brain. So you see food, you smell food, you think of food. Or in this case, you have been trained to hear a bell and expect food. So that eventually, hearing just the bell created an association, caused salivation and acid secretion, even though no food was ever actually administered. You can also get acid secretion in response to sham feeding, where food can get in, you can taste it in the mouth, but it doesn't actually arrive in the stomach. So there's no trigger, no food or anything arriving in the stomach to cause a reflexive reaction to get acid secretion. It's all neuronal, mediated by the vagus. So, when we're looking at your three phases, on the top you have the cephalic phase, that's via the vagus, okay? The gastric phase, these are local reflexes. You have not just acid secretion because of the presence of gastrin and histamine, but you also have what's called vagovagal reflexes. Stretch receptors in the stomach, by adding food, will trigger a reflexive reaction. You stretch and you get the response to promote secretion from parietal cells because now you have something in there that needs to be digested. The intestinal phase is going to be hormonal but also nervous mechanisms. So the first figure that I have, the gastric is just basically summarizing the same part, except we're looking specifically at distension. So that sensory input will go into the enteric plexus and will result in effector cells that increase secretion and motility. So long reflexes that are coming all the way to and from the brain, or a short reflex that's coming just from the enteric nervous system. These local reflexes and vagovagal longer reflexes are going to involve two specific hormones. Or not hormones, sorry, neurocrine secretion compounds. You have acetylcholine and you have GRP. What is GRP? Gastrin-releasing peptide. Okay, so it tells you exactly where its target is and what it's going to act on. So your antral G cells are going to respond to GRP. They also can respond to acetylcholine. Acetylcholine will act on G cells, ECL cells, and parietal cells. So it's going to go and bind to M3 receptors on all three of those cellular targets, and it's going to be coupled to G proteins. Okay? Histamine, gastrin, and the actual parietal cell itself are all going to have an excitatory signaling cascade that will result in the release of their either mediator, their hormone mediator, or directly for acid secretion. Somatostatin is also going to bind to a GPCR, but it's going to be coupled to which type of G protein? Inhibitory, a GI protein. So in this case, we have food that's present in the stomach. Specifically, if we have proteins, they can be broken down into peptides or amino acids or peptones, all of which are going to be neuronal or the presence of food in the stomach. Okay, so you're at your gastric phase when you actually have food arriving in the stomach. It can be neuronal by stretch reflexes that promote secretion and motility, or it can be a chemical response because specific types of nutrients are sensed by the surface epithelial cells inside that corpus region. All of these are going to result in parietal cell promoting secretion of acid. This was the figure that I preemptively mentioned. So if you're looking at the lumen of the stomach, the cephalic and the gastric, the integration of the two of them is shown in this figure. We have your input coming from the vagus, so this would be involved in your cephalic phase. This is something that whatever sensory information tells the brain that food is coming to the stomach will not only promote salivation, but it also promotes a preliminary acid secretion and gastrin release from the stomach, okay, to prepare for food coming in. It's a more efficient way. It's sort of laying the groundwork so that when food gets there, everything is already up and running. The gastric phase is what happens when the food actually gets into the stomach, okay? So in terms of acid secretion, we have two different sections of digestion that are going to be responsible for the release of acid. And these two together are responsible for like 90% of acid being released. 30% of it is going to be due just to the cephalic phase. 30% of all your acid secretion is strictly neuronal, just from sensory information from the idea that food's going to arrive in the stomach. 60% or so is coming from food actually getting into the stomach and then the stomach responding to the presence of food. Now, the second that you get all of these mediators being released in order to promote acid secretion, you're also triggering somatostatin release so that your acid secretion doesn't go haywire. It's an instant, simultaneous release of mediators for secretion and mediators to inhibit that secretion. The intestinal phase. The intestinal phase is really going to be responsible for getting all of your nutrients down to an absorbable size. So typically, what is happening in terms of acid secretion from the intestinal phase? Are you stimulating or inhibiting usually? You're usually inhibiting, right? Because once food gets into the intestine, it's already been digested enough to get small enough to be able to fit through that 2-millimeter gap in the pyloric sphincter. And if you have enough mechanical and chemical digestion to produce that chyme that can get through, now we don't really need as much acid being released. So what's going to happen is, in the lumen of the small intestine, it's going to trigger all of the digestion and then the release of those pancreatic secretions to promote enzymatic chemical digestion. You still get mechanical digestion in the intestinal phase. It's that segmentation of the intestines that sort of churns it around and helps to disturb that unstirred water layer. But the bulk of when we are talking about chemical digestion, most of it's happening in the small intestine, which is why we're skipping over lecture 20 content, because there's a lot going on in the small intestine. And all of these pieces we're going to get into in just a little bit. When you have protein digestion products showing up in the intestine, you can get some stimulation of acid secretion. It's relatively minor. It accounts for maybe 10% of all the acid secretion, okay? Because it's going to happen as a way to just sort of finalize the last little bit of digestion happening in the stomach, but you also have that same response to the presence of acid to shut down acid secretion. So it sounds a little counterintuitive to think that the intestinal phase, where you have food arriving in the small intestine, is actually promoting acid secretion, but it does in very small quantities. It's not a very potent stimulator. It is a more potent stimulator of somatostatin release. So it's sort of the tail end of any acid secretion. But through a series of studies, there is evidence to show that protein digestion products, specifically amino acids, can trigger the G cells that are present in the intestine to release big gastrin. And if gastrin are released in response to the presence of peptides and amino acids, you're going to get a modicum of release of acid in response to that. Okay? Don't worry about enteroxantin. That was a name that they came up for a mediator that's been identified but hasn't fully been sequenced. They don't really understand the exact mechanism of it. And so entero meaning intestine, oxinin meaning the oxinic gland of the oxinic region of the stomach, which is the parietal cell corpus region, to release acid. So that's where the name comes from. But I don't expect you to memorize that, but I wanted to explain where that comes from on the picture here. So this is from the boron book. So there does appear to be another mediator that seems to help augment acid secretion aside from big gastrin, but it's not fully understood yet. All right, the last part I want to talk about is specifically when we were talking about how you can get acid secretion from peptides arriving in the intestine, is mainly because what products are getting digested in the stomach? Proteins, fats, and anything else? Carbohydrates from salivary glands, right, from saliva. But does that continue in the stomach when you get to the acid level? Not so much. I mean, it will still continue to function, but at a very low level. That's why you also need pancreatic amylase to continue that further luminal digestion of carbohydrates. But you don't get any protein digestion until you get into the stomach. Pepsinogen is the first stop for protein digestion. Okay? It's released by your chief cells, and it is not something that has to be cleaned. It's released as a zymogen, which is good because you have a lot of proteins on the surface of your cells. So if your proteases were released as active enzymes, it would digest your cells. So released as a zymogen. What triggers its activity? Acid, pH. A drop in pH activates and converts pepsinogen into pepsin. Okay? You have to get that pH down to 3.5 or lower. How low does it get at peak acid secretion? Certainly less than 2, a lot of times less than 1. Okay? Massive amounts of acid secretion in response to food is very sufficient to get activation of pepsinogen into pepsin. Okay? As long as it's below 3.5, you're going to activate pepsin. So what is the pH of the stomach normally at basal levels with your baseline secretions? Is it neutral? Is it acidic? Is it alkaline? Anybody brave enough to say it a little louder? Because I'm getting a lot of mumbling. Slightly acidic, 4 to 6. So not at the level where you're actually going to have pepsinogen being activated to pepsin. Okay? So when you're not eating, even though your stomach is still acidic on the regular pH scale, it's not sufficiently acidic in order to get pepsin activation. What happens if it gets to more alkaline, above 7.2? What happens to pepsin? It's irreversibly inactivated. So between about 3.5 and about 7.2, pepsin is reversibly inactivated. So any pepsin that's floating around that was activated, once the pH goes back up above that 3.5, it'll become inactivated, but ready to be activated again as soon as the pH drops. If you chow down on antacids and you actually create an alkaline pH in your stomach, you can irreversibly activate whatever pepsin is around in there. So the only way that you would ever be able to get protein digestion in the stomach is if your pH were to drop and more pepsinogen were to be released, that could be activated once the pH gets sufficiently low. Okay? All right. So we have a layer that protects your epithelial cells that line the stomach. So not only are we looking at the chief cells that we're talking about releasing your pepsinogen, we have your parietal cells that release intrinsic factor and your acid. What are the other cell types that are going to be triggered to release something to protect the lining of the stomach? Do you remember what you have neck cells and surface mucus cells. You're going to be releasing not just mucus though. It's also a very bicarbonate-rich solution. And that mucus has bicarb being released alongside it, so it creates this neutral or much more basic coating around the epithelium. So one of the things that does, you have a higher pH, it protects against the acid, right? What else does it protect against? What's activated in response to acid? Pepsin. What happens if pepsin somehow makes it through that and into that mucus bicarb layer? It could, in theory, digest your cells. If your pH is high enough to make sure that it's irreversibly inactivated, it's a way of protecting your stomach cell line. Okay? So this layer is really there, even though you get release into the milieu inside of your stomach for activation, it also protects those cells so that if it ever does come back into contact, pepsinogen will be, or pepsin will be inactivated so that it doesn't auto-digest your cells. Okay? So this is what it really looks like if you wanted to kind of zoom in. This little square here on the left shows your normal epithelium, and if you zoom in on that, you're looking at a pH profile. So normal physiological pH, 7.4. Once you get into crossing over the apical side of the stomach, you're looking at more of a neutral pH. So it does decrease as you get past that mucus gel that is laden with bicarbonate. The neutralization zone is really just the contact point that's sort of that mixture region. It's not a hard and fast line where you have pH of 7, pH of 1, right? You have a little bit of mixture that's happening. So you'll have some protons coming in, some bicarb that's exiting, but it still provides this really good, solid pH and physical mucus barrier, okay? So the only way that these cells can continue to survive and do their job is if they're protected from all the stuff that they're secreting. Yeah? So in theory, that could contribute to it, but if you hold on just a second, I will explain where stomach ulcers come from. Yeah. Because this is not technically right at that pH of above, it's got to be above 7. This is just below that value. So it's reversibly inactivated. If you had excess amounts of bicarb, it could, in theory, inactivate some of it. So when we say that pH has to be a 7.2, it's not really a magic number. It's sort of, that is the average. Some of the pepsin will irreversibly inactivate maybe at a pH of 7. Some of it won't irreversibly inactivate until 7.5. But it's sort of within that error margin. So this is there to make sure that you still have most of your pepsin that's reversibly inactivated, but you have to have a significant pH drop before you actually get any kind of activation. But some of them will be irreversibly, but you're constantly getting pepsinogen being released, And the probability really is looking at the overall numbers. The amount that's getting released and the amount that might potentially be irreversibly inactivated is relatively small. Yeah?
Do proteins themselves cause the acidification, or do they indirectly cause the acidification?
Say it one more time.
Do proteins themselves directly cause acidification, or do they indirectly cause it? Because not all proteins are acidic. So do they indirectly cause the acidification to trigger?
So the acidification has to do with the sensing of any nutrients and the release of acid to acidify. It's not actually looking at acidic amino acid side chains.
Okay.
All right. I already covered that. All right, so a little bit to talk about your ulcers. A couple ways that you can damage the integrity of your mucosal barrier. If you want to strengthen it, you need to have what we talked about already. You need to have mucus. You need to have bicarb secretion. Gastrin can also strengthen the mucosal layer. How would gastrin strengthen the mucosa? What else does it do besides cause parietal cells to secrete acid? It causes proliferation of those same parietal cells, right? It stimulates growth factors, okay? So by stimulating those growth factors, you can get more increase in cell replication, reproduction, and either increase the total number of those cells or at least increase cell turnover. There's also epidermal growth factor that will help to release, stimulate the proliferation of mucosal cells as well as prostaglandins. So the barrier would be healthy and you wouldn't end up with ulcers. If you want to weaken it, aspirin, NSAIDs, ethanol, all of these things are, for lack of a better word, toxic to your epithelial cells. They weaken the overall lining. They damage the integrity of those epithelial cells, increase the chance that they will go under apoptosis to be able to be recycled. Bile salts could do that too if you have some bile salts that are going to come back up in. Mostly bile is going to end up going straight into the intestine. But the one I really want to talk about is H. pylori. How many people know what Helicobacter pylori is? You've heard of it. Do you know why you've heard of it? Do you know the name Barry Marshall? How many people have heard of him? Who knows the story? Anybody want to tell it? Go ahead. Yes, he did. So this is your press release from when it happened. Barry Marshall couldn't get approval for conducting his experiment. So what he did was he collected H. pylori from patients that had gastritis. He purified it, he grew it up in a culture, and he drank it. And he tested it on himself as a proof of concept, not recommended. The only reason he started taking antibiotics was because his wife had basically had enough of his complaining about all of the horrible symptoms that he had. He had an endoscopy before he took the culture to prove that he did not have any peptic ulcers. He didn't have gastritis. He took this and then he monitored himself. And the end result was this lovely publication that says here, the subject was one of the writers, Barry Marshall, a 32-year-old man, a light smoker and social drinker who had no known gastrointestinal disease or family history of peptic ulceration. He developed it immediately after drinking this solution of H. pylori. It took hold, it created ulcers, he had multiple endoscopies to prove the progression of this, and then when he took antibiotics at the behest of his wife, who was getting frustrated, decided to end his study before he was planning to end it, and found that it resolved. So how many people have taken microbiology? You've heard of Koch's postulates? Koch's postulates are 4 criterion that were developed by Robert Koch to determine that a microorganism caused a disease. And if you can fulfill all 4 of these, then you have proven that this microorganism causes this disease. That's what he was attempting to do here. And obviously he did it, and he won the Nobel Prize for it. But it's not a great lesson in research ethics, but it was obviously done many, many years ago. This publication came out in 1985, and it was in the Medical Journal of Australia. So, interesting story. Not recommended for any of you to actually do anything like this when you do research, but it did prove that anytime somebody comes in with gastritis or peptic ulcers, it's a good idea to give them a test and see if they have bacterial infection. Give them antibiotics and see if it clears up. The acids can contribute to it, because once you get peptic ulcers, you're losing the ability to provide that lining, and the acid will continue to have access now. But it's not really the initial cause of all of those ulcers. Okay, so that is the end of our lecture 19 content. And now we're going to get straight into our intestinal stuff. So first, a little bit of anatomy. Oh, yeah. Yeah. How would it affect their digestion? So, in what way? So, it's not so much that it's necessarily going to slow down your digestion as it's going to cause damage to the lining, which, if long-term and prolonged, could damage the mucosal barrier in your stomach, which means it's damaging the cells that are releasing intrinsic factor, releasing your acid, releasing pepsinogen. So it can slow it down, but if you lose the ability to release pepsinogen or activate pepsinogen, it doesn't actually result in a loss of protein digestion. Your pancreatic enzymes are more than capable of maintaining complete digestion of your proteins. So it can slow it down, but only if this is something that's chronic, because you do have a very high turnover of the epithelial lining anyways. The big problem is that if you have these peptic ulcer diseases, that damage can also, depending on where it occurs, if it occurs up toward the lower esophageal sphincter, you can start getting more reflux, and then you can end up with something like Barrett's esophagus. Yeah. Mm-hmm. Yep. Yep. Cortisol is another example. All right, so things that we're going to talk about. We're going to focus a lot on your carbohydrates, proteins, and lipids. That's mostly what we're going to do in the beginning today. We'll talk a little bit about bile acids, how they're related to the breakdown of fats, and then we're going to get into cobalamin, which is happening to get absorption in the ileum. But that probably won't be until Thursday. All right, so how do we get absorption in the small intestine? We have a massive amount of surface area, okay? You're talking 250 to 400 meters squared. That is a lot of surface area. The folds of Kerckring is really a threefold increase. So the small intestine, first of all, it weaves around like a ride line at Disney World. Okay, it's compacted in there, but it's so turned back on itself. There's a lot of surface area just from pulling it out. Okay, our small intestine about 4 meters long. It's further broken down into more surface area by the folds of Kerckring, which is just infolding from the actual small intestine. Then your villi, and then your microvilli. All of these are going to really ramp up the total surface area. The more surface area, the more cells there are that your food products can have contact with for digestion. So this is really what your folds of Kerckring look like. They're also called valvulae condimentes, but folds of Kerckring is a little bit easier to remember, and it gives credit to whoever discovered it. So these are your folds. On top of them are the villi, and then on top of those are your microvilli. So the villus itself, you have the central lacteal, and you have blood capillaries along with your arterioles and your venules that are going to feed that particular villus. So every villus is those finger-like projection coming off of the surface of your epithelial lining of your intestine. But each one of those villi are covered by microvilli. This is also called your brush border because it looks like a bunch of bristles. This will be really important specifically for the digestion of products to an absorbable amount. The ones that are released from the salivary glands, released from epithelial cells inside of the stomach, and released through the pancreas, are acting in the lumen. These are luminal enzymes, and they don't give you monosaccharides, and they don't often give you individual amino acids. Brush border enzymes are going to be critical for that final step to break it down in order to get it across the barrier and into your bloodstream. Okay? So brush border enzymes will reside right on that microvilli layer. This is really a summary just to show what it is that we are going to be talking about. Starting with our glucose, we have our different disaccharides, proteins and amino acids, peptides and fatty acids, how all of them are going to have to get through an enterocyte. So the epithelial cells in the intestine are enterocytes. Entero for intestine. So when we talk about enterocytes, we're specifically looking at the surface absorptive epithelial cells. All of them have to pass through in order to get absorbed into the bloodstream so it can go to different parts of your body. This is not to say that everything has to be processed inside the cell. Some of them will be passing directly through, but it's not a paracellular movement. Paracellular movement is reserved for water and ions, okay? Not for small molecules like nutrients. There are many barriers to get absorption across that epithelial lining. We have a nice little list here that shows 8 different barriers by which a nutrient has to get through to get to the capillary wall or the lymph vessel. You have that unstirred water layer, which is going to be disrupted by segmentation inside the intestine in order to basically pull off that unstirred water layer and expose the apical layer of your cell. You have a glycocalyx. So at the beginning of the semester, we talked about that glycocalyx, those surface carbohydrates that are attached to the apical side or the outside of your cell membrane. Then you have the actual cell membrane itself. Then you have the cytoplasm of the enterocyte, the basolateral cell membrane. There's an intercellular space between the basolateral membrane and the basement membrane, which separates your epithelial lining from the endothelial lining and the connective tissue. Then you have the wall, the endothelial cells of your capillary membrane or your lymphatic system before you actually can get into your circulatory system where these nutrients can go to different parts of the body. So lots and lots of layers to get through. Only the membranes themselves are going to be major barriers for allowing specific types of nutrients to get in, as opposed to being restricted based on size. So we're going to start with carbohydrates. We're pretty much going to eat some monosaccharides, but a lot of oligosaccharides and polysaccharides. Okay? Most of what you eat is starch. Digestion of starch does not produce monosaccharides. At most, it produces disaccharides and oligosaccharides. Okay? We also will ingest some non-digestible polymers. You can have pectins, which are more easily digested than cellulose by bacteria that are present in the intestinal tract. Lignans don't. Lignans are like wood. It's an aromatic polymer that will function as a fiber source but doesn't actually get digested. So it's really there just to kind of, once you eat that, it will help with motility, but it doesn't provide any kind of nutrient. To get down to glucose and fructose, it's a very small amount that you're actually ingesting. So we have to take your starches and break them down into monosaccharides before they can ever cross into the enterocyte. How do we do that? You have alpha amylase and you have pancreatic amylase, both of which are luminal enzymes. They're going to cleave those alpha 1,4 linkages between all of these monosaccharides. But they cleave internal bonds. They don't cleave bonds at the end of a chain. So you can't have any monosaccharides resulting from amylase digestion. Since it can't cleave a terminal bond and it can only cleave interior, the most you can get is a disaccharide. And we need monosaccharides to be able to cross into the body. Okay? So at most, you get maltose, which is what? What's maltose? Biochem 451. It's disaccharide of what? Glucose and glucose, right? Sucrose is going to be glucose and fructose, and Lactose is glucose and galactose, okay? So maltose is your glucose and glucose disaccharide. You can have maltose and you can also have glucose polymers in smaller numbers, so 3 to 9 glucose molecules together. Those will be broken down by maltase and alpha dextrinase. That will produce glucose. I'm going to show you on the next slide. This one's included because it's a little bit simpler. The next one, I believe is the next slide, comes from the Boron book, and it's kind of busy, but it gives you a lot more of the details that we're going to talk about. The glucose is going to be the end product here of digestion by maltase and alpha dextrinase. Lactose and sucrose are also potential products of starches. Lactose will be digested by lactase, and sucrose by sucrase. The end result is that you're going to get lactose broken down into galactose and glucose, and sucrose will be broken down into fructose and glucose. What is this blue area, that box? What do you think that represents? The brush border. Okay, that is the area where we already pointed out before when we looked at the electron micrograph, all those microvilli. These brush border enzymes reside in those microvilli. Okay? These enzymes are critical to get monosaccharides so that they can actually be absorbed by the enterocyte and used by the body. This is the one I was telling you about. Okay? Kind of a busy figure, but the thing that it does a really good job of doing is it shows you the different types of linkages that are broken down. Alpha amylase is showing an amylose and an amylopectin. What it's showing is that you are cutting the alpha 1,4 linkage, but the internal alpha 1,4 linkage. That's what's shown up here on the left. You can't get a terminal alpha 1,4 link. You can't get a branching 1,6 linkage. That's this guy right here. Any kind of 1,6 linkage is not going to be able to be digested by amylase, even if it's an interior one. And because of the presence of these in a variety of starches, the alpha 1,4s that are in between or immediately adjacent to that branching 1,6 linkage also can't be digested by amylase. So amylase's job is really just to chop up your starches into a bunch of smaller pieces so that your brush border enzymes can break them down into individual amino acids. Its only ability is to cut alpha 1,4 linkages of those monosaccharides, and only if it's internal and as long as it's not right next to one of these alpha 1,6 branching linkages. Okay, so lots of stuff that can't be cleaved by amylase. When you look over here, we have our lactase, we have glucoamylase, and we have the sucrase and isomaltase, all of which are going to help break down these alpha limit dextrins, the ones that are incapable of being broken down into monosaccharides from amylase. Okay? So the isomaltase is the one that's going to be able to help break down the terminal monosaccharides to break apart not only individual monosaccharides, but it can cleave this alpha 1,6 branching linkage as well. Okay? So all of these are going to reside alongside the transporters on that brush border so that monosaccharides are released and can immediately bind to whatever the channel is that they're going to be utilizing. Does anybody have questions about this so far? Okay. All right, so most of what we're going to have that is broken down is glucose. We will have some fructose, we will have some galactose. There's a big difference in the way that glucose, galactose, and fructose are all going to be absorbed. And it has to do with the specific type of transporter or carrier protein that is going to mediate the absorption on the apical side. Glucose and galactose use the same transporter. Most of what you're going to eat is glucose, so it's going to be able to be taken up more readily. But galactose uses the same transporter. So they're sort of competing for binding to that SGLT1 protein. The SGLT1 is a secondary active transporter. You are moving these sugars against their concentration gradient to get it taken up by the enterocyte. What are we going to use for the energy source? Sodium. Okay? Sodium moving down its gradient into the cell is going to bring along glucose and, to a lesser extent, galactose. Fructose is not going to require energy. It is going to utilize its concentration gradient, and it's also going to utilize a totally different transporter. GLUT5 is how fructose gets across the apical membrane. Based on your biochemistry, whether you took 451 or not, what is the difference in the molecular structure between glucose and galactose versus fructose? Yes. 2 are Pyranose rings, which means 6-membered rings. Okay? They all have 6 carbons, but the question is, what is the three-dimensional formation? Your glucose and galactose form a 6-membered ring. Fructose is a 5-membered ring. Okay? And it turns out that in order to get SGLT1 to work, you need a 6-membered ring. So this is just the model to show what we had already talked about, where you have SGLT1. It's going to move sodium and glucose or galactose coming in on the apical side, and then your sodium will be pumped out from the sodium-potassium pump, and your glucose and galactose will diffuse by GLUT2 on the basolateral side along with fructose. So basolateral, they all use the same transporter. But on the apical side, only glucose and galactose can use the SGLT1. And that's basically what's happening here. You have SGLT1 and you have GLUT5 that are on the lumen of the intestine. All of them, all of your nutrients, no matter what type of ring it has, can utilize GLUT2 coming across the basolateral side. Okay? But they need different apical transporters. Okay? So some, for those of you that did endocrinology, did anybody talk about expression in pancreatic islet cells for release of insulin and how glucose triggers that? Did they explain why fructose doesn't typically trigger it? Why fructose doesn't typically trigger insulin release, but glucose does? The GLUT4 is for glucose being able to come in, but it doesn't express GLUT5. GLUT5 is specific for that 5-membered ring of fructose. And if it doesn't get absorbed into pancreatic cells, it can't trigger the release of insulin. So that's one of the reasons why your dietary fructose doesn't have the same signaling cascade in terms of your body's response to sugar that glucose would. Even though they can all use the same one on the basolateral side, you have GLUT1, 2, 3, 4, and 5, and they all function somewhat differently as well as in different parts of the body. Oh, sorry. So that was just to point out that all of them use the same basolateral side. So this figure is just the exact same information from the boron book. SGLT1, this is really what it looks like. I'm not going to ask you to memorize this, but you should know that it is a very extensive transporter, and the big take-home message from it is that you have to have a Pyranose ring, and it has to be in the D configuration. The only way that you get amino acids absorbed in your body is in the L configuration. The only way that you get monosaccharides absorbed is in the D configuration. The SGLT1 will not absorb any L-glucose or L-galactose. It has to be in the D configuration, and it has to be a Pyranose ring, which is why it also cannot move fructose. This had more to do with what I had just mentioned before. Insulin release can promote leptin release, which causes you to stop feeling hungry. This is just a physiological consequence of the difference between glucose and fructose. If fructose can't trigger insulin release, you tend to get less leptin coming in, so it tends to not be as responsible for being able to suppress hunger and promote that feeling of fullness. So there's a little bit of an argument about exactly what the role of the liver is in terms of forming a lot of the fats that would explain why you would have high fructose corn syrup playing a big role in obesity. There's a lot of conflicting data going around, but the take-home message is that your body doesn't respond to fructose the way it responds to glucose. Glucose is sensed, and the insulin that's responding to it not only responds to the breakdown of it, promotes the usage of glucose by cells, but it also triggers the release of hormones responsible for feeling full and stopping that feeling of hunger. That kind of normal feedback mechanism isn't introduced when you consume fructose. And our normal diet isn't fructose heavy, right? That's a manufacturing thing because it's a lot cheaper to put fructose in, and fructose is a lot sweeter by taste per unit. It tastes a lot sweeter, so it's less expensive to use that to sweeten things. So take-home message, the different transporters for the apical side of how you get absorption of these two coming in, they can all use the same basolateral, but if you have a defect on the apical side for one of those two transporters, you will affect the ability of enterocytes to absorb different types of monosaccharides. Okay? So amylase breaks down all your starches, but your brush border enzymes are critical to get any kind of absorbable nutrient that's a carbohydrate. All right, any questions on carbohydrates before we go into protein? Okay, so proteins. Same kind of things going on, except you don't necessarily have to have just amino acids in order to get absorption. You can have dipeptides and tripeptides. The big take home here is that you still have to have something that is very, very small. The difference is it's not the same in your carbohydrates where you have to have a single unit. You can have two amino acids linked together, and they can be taken up across the enterocyte apical membrane to be taken up into the body. Now, the big transporter is your peptine-1, okay? All of these proteases and pancreatic peptidases are going to break down those proteins so that they can either be absorbed by individual amino acids, and that's going to be your sodium amino acid transporter. Same mechanism or same energy requirement that you have for the glucose and the galactose for your SGLT1. What happens here is it's going to use your sodium gradient to fuel the movement of those amino acids. Peptine-1 is your oligopeptide transporter. It can move your di and tripeptides. Okay? So small peptides can be moved across the apical membrane, and they can either be transported directly across, or they can be digested inside of the enterocyte to produce amino acids. Okay? Your protein digestion also relies on brush border enzymes in order to further break down any larger peptidase or peptides that were not broken down by the different peptidases and proteases. So we have a variety of luminal proteases, and then we have our brush border proteases. So pepsin takes your proteins and breaks it down into proteopeptides, polypeptides, peptones. Then you have your trypsin, chymotrypsin, all of these that are going to be released in the intestine with the pancreatic proenzymes to allow for polypeptides and amino acids. Then you have peptidases that are at the brush border to get your individual amino acids. Okay? So same principle where you have some that are getting digested in the lumen, and then you have that final step of digestion happening right at that microvilli border. You remember we talked about enterokinase and we talked about enteropeptidase being the same thing? This is looking at enteropeptidase, which is going to cleave trypsinogen into trypsin. So, what else does trypsin do? Not only does it activate a lot of other enzymes, but what else does it do? It activates trypsinogen. So, enteropeptidase is an enzyme that is present at the brush border that can activate trypsinogen, which can then further activate itself. It's autocatalytic. Enteropeptidase has also been implicated in stimulating growth factors to promote cell turnover inside of the intestinal lining as well. But we have trypsin, chymotrypsin, and our elastase, as well as carboxypeptidase A and B. Now, I haven't mentioned those yet, but it really has to do with what the purpose is of each of these types of enzymes. So even though we've talked about pepsin and we've talked about some of the proteases, each one has a specific goal. You have endopeptides, or peptidases. Your trypsin, your chymotrypsin, your elastase, all of them are going to cleave amino acids at different sites. It's cleaving the whole protein, your whole polypeptide, at different locations. Trypsin is going to cleave at your basic amino acids. Your chymotrypsin is going to cleave at your aromatic amino acids, and your elastase at your neutral amino acids. These are endopeptidases, which means they're cleaving which type of bonds? Internal. Okay? They're cleaving internal bonds of that polypeptide. Okay? Your exopeptidases are cleaving individual amino acids. The endopeptidases will not give you individual amino acids. Okay? Why do we need all of these? We need them so that you can get individual amino acids showing up either at the aromatic and neutral aliphatic. So aliphatic is your linear chains, your alkanes, alkenes, alkynes. And then your basic amino acids for carboxypeptidase B. The biggest difference between these two, internal bonds versus external bonds, endopeptidases are about 70% of all of your digestion. The last 30% of your protein digestion occurs from the exopeptidase. And the reason you don't need to have the exopeptidase cleaving every single one to an individual amino acid is because there are avenues for di and tripeptides to be absorbed. Okay? So this just kind of summarizes what we have here. Breaking it down into large peptides, brush borders are going to break them either into free amino acids or down into di and tripeptides. The difference is two different sets of carriers. Two different sets of carriers get them across that apical membrane. Once inside the enterocyte, they can be broken down into individual amino acids for movement across the basolateral side. But you have to get it down to single amino acids or di and tripeptides because the transporters are there to be able to move them across, require either an individual amino acid or a very small peptide. One thing that I want to point out is that sometimes there seems to be a kinetic advantage to taking up a di or tripeptide. And it might have to do with the type of transporter that's used for the peptides versus for the individual amino acids. So it's a proton-driven co-transporter across the apical side, that PepT1. It showed that the appearance of glycine in the blood happened faster if you had two glycine amino acids as a dipeptide than if you gave individual glycine amino acids in this experimental design. So it suggested that there might actually be a way to improve the uptake of proteins, or I should say of amino acids, by having them as a very small peptide. But it's most likely because of the kinetics of the different carriers that are utilized. Whole protein uptake, very important for babies, not so much for us, at least in terms of digestion. Infants will take up whole proteins. It's also the basis for the mucosal immunity that they get from the passive immunity from feeding. For us, it has more to do with the immune system. We don't really get nutrients from whole protein uptake. We break down all of our proteins and take them up as amino acids or small peptides. Whole protein uptake does happen. You get phagocytosis of an entire protein either by an enterocyte or more likely by an M cell in the Peyer's patches that's part of your gut-associated lymphoid tissue. And the purpose of that is to generate antigens that you can present into those immune cells that are right on the other side of that intestinal lining to allow for your immune system to respond to anything that you might have eaten that would cause a reaction. Okay? I think I've mentioned this before, that your immune system has to be able to respond to toxins and pathways, that would be introduced to the body through an epithelial lining. Your lungs have direct contact with the outside, so you have a lot of irritant receptors to prevent stuff from getting to the alveoli. Your gut has its own really extensive immune system response and a way to process antigens so that it can prevent those from getting further into the body. What happens if you don't have normal protein assimilation? There are a lot of different physiological, or I guess pathophysiological effects that can be from a lack of the ability to assimilate protein. So assimilation of any kind of nutrient, we're talking about digestion and absorption together. That's what your assimilation means here. Pancreatic insufficiency, if you don't have pancreatic proteases, you lose a lot of your ability to digest proteins. Most of your proteases are going to come from pancreatic secretions. So you'll get a lot of nitrogen ending up in the stool, and you won't have as much being absorbed. Normally, less than 4% of all the nitrogen that you consume actually shows up in the stool. Most of it gets processed and reabsorbed. You could also have somebody who has a congenital absence of trypsin. If you don't have trypsin, you don't have any of the other proteolytic enzymes. The most you can do is breaking down with pepsin. And you'll get some digestion, but only if you're lucky enough to get a few ditripeptides can you get a little bit of absorption of proteins. Heart disease is one I wanted to specifically target because it's an inability to absorb neutral amino acids. So it doesn't mean that you can't get any of those essential amino acids. It just means that you can't absorb them as individual amino acids. So one of the things that can be done is this was how we sort of demonstrated that there is a separate carrier mechanism for di and tripeptides compared to individual amino acids. If you were looking at arginine, which is charged, and phenylalanine, which is not, a normal subject would absorb all of them at the same relative rate, whether they were individual amino acids or whether they were a dipeptide. Somebody with heart disease has a problem with the phenylalanine being absorbed because they have no transporter that can absorb neutral amino acids. They have no problem with arginine. So in this example, you see that the red lines, you have normal arginine absorption, but you have very, very low phenylalanine. However, if you gave it as a dipeptide, you didn't have a problem. So it doesn't mean that a defect in one of these amino acid transporters means you don't ever have that amino acid. It just means that it will likely be requiring the use of a functional peptide-1, so for di and tripeptide. So this was solid experimental evidence that showed we do have the ability to take up peptides and not just individual amino acids. All right, I'm going to stop there, and we'll finish up lipids and cobalamin on Thursday.